Good evening to all. I warmly welcome for today's CellTalk webinar. Today's session is going to presented by Shruti and the research paper titled Genetic Networks of Alzheimer's Disease, Aging and Longevity in Humans. Our speaker Shruti is working as a graphic designer intern at Renix Life Sciences. She is responsible for designing various user-friendly designs on healthcare-related topics, focusing mainly on gene-based disease projects, FEMED field program project, website interfaces, and logos. Shruti has a bachelor's degree in life sciences from Osmania University, Hyderabad, and her designing work focused on healthcare-related topics, magazines, brochures, flyers, pamphlets, social media images. She has gained expertise at Renix not only in UI/UX designing, but also in the certain form of aesthetical pleasing user interfaces, time management, and information organization. Without further delay, I would like to invite Shruti to take over the session. Over to you, Shruti.
Yeah, thank you, Renuka. Good evening, everyone. Today, my CellTalk presentation title is Genetic Networks of Alzheimer's Disease, Aging and Longevity in Humans. The original author was Balmeros T. et al. Yeah, the title is Genetic Networks of Alzheimer's Disease, Aging and Longevity in Humans. The topic is selected from Longevity, and the journal title is International Journals of Molecular Sciences, which has an impact factor of 5.6, which is published in the year 2023. This research has an impact on longevity, aging, and Alzheimer's disease. Yeah, when it comes to introduction, the genes which was associated with the early and late onset of Alzheimer's disease, that is AD genes, they have been discovered using human genome research and also the genome-wide association studies, which is also known as GWAS. The Alzheimer's disease is the most frequent cause of dementia, in which about 5 million people were living with AD in 2014, and the number is estimated to nearly triple by 2060. Aging and the genetic variations are well-known risk factors for the Alzheimer's disease, and also the vast majority of genetic risk factors fall into late-onset Alzheimer's disease, that is low. As you can see in the picture, the Alzheimer's disease, the presence of amyloid plaques in the presence of neurons, it indicates the disease of Alzheimer's disease, that is dementia. And this is a normal neuron, which is a healthy one. Yeah, when it comes to the general background, the previous studies were focused on specific set of genes that has been shown to contribute as the risk factors for Alzheimer's disease. Thus, the connections among the genes involved in Alzheimer's disease, aging, and longevity, they were not well understood. Hence, they identified the genetic interaction networks that referred as the pathways of aging and longevity within the context of AD genes by using a gene set enrichment analysis that is known as Reactome, which is a bioinformatics database tool, and also which cross-references more than 100 bioinformatic databases that allow the interpretation of the biological functions of gene sets through a wide variety of gene networks. Yeah, when it comes to the present challenges, genetic hallmarks identified provides the unexpectedly broad mechanisms suggesting a wide variety of implications in the field of aging. Alzheimer's disease genes, they create the ground for age-related comorbidities, particularly mitochondrial RNA metabolism, cytokine signaling, and also the lipid metabolism with the highest significance. The Alzheimer's disease genes, they were associated with the common age-related comorbidities, which includes diabetes, myocardial infarction, heart disease, hypertension, cardiovascular system disease, and also the vascular disease. From this, I can say that there is an inverse relation between the Alzheimer's disease and cancers, because the patient who was suffering from cancer, they have the low chance of presence of dementia. Yeah, when it comes to the objective of the research, the reactome analysis used in this study that provides the genetic pathways with the gene set enrichment analysis and also the statistical confidence level. Yeah, when it comes to methodology, the materials they use three different types of data sets along with the overlapping data sets and also the gene ontology, which is the reactome analysis. When it comes to three different types of genes, they include the human Alzheimer disease AD genes and also the AR genes. Those were the aging related genes and also the longevity genes. And the two different additional sets were created based on the previous one. That is, first they identified the genes which were shared by both the AD genes and also the AR genes. They were considered as AD and AR overlap. And also the second one, the identification of the genes which was shared by both AD genes and longevity genes. They are AD and longevity overlap. When it comes to gene ontology, this is a reactome analysis which is completely based on app. That is Reactome Physis, which was used to determine the enrichment in the functional interface network. They use the cytoscape, which is the version of 3.8.2, that is a Java version, 11.0.6, to run the Reactome software plugin, that is Reactome Physis app. Yeah, when it comes to methodology, as I said, gene ontology, which is the Reactome, it is an open source bioinformatics database, which consists of two functions. The first one is the database comprises of 27 groups of biological pathways. They include the autophagy, which is self-eating of intracellular cells, and also the cell cycle, cell-cell communication, cellular responses to external stimuli, chromatin organization, circadian clock, which is present in the brain, that helps the individuals to alertness and also the sleep-wake-up cycle, and also the developmental biology, digestion and absorption, and disease. The pathway reaction and the molecule pages were extensively cross-referenced to more than 100 bioinformatic resources, including the chemical entities of biological interest, small molecule database, ensemble and uniprot database, NCBI gene, and also the UCSC genome browser along with the PubMed. It is manually curated and also the peer-reviewed knowledge base of biological pathways. This is the first function, and the second one is gene set enrichment analysis, GSEA, that can be carried out using it as a bioinformatics tool. GSEA is a computer technique that uses genome-wide profiles to determine and interpret the biological activities of a group of genes. Yeah, here, as you can see here, there are three different types of databases. First one is the Allgene database, which is from Alzheimer disease. And this is the GeneAge database, which is from aging related genes. And this is the longevity map database, which is from longevity genes. They are based on these three databases, they perform the reactome analysis and they get the overlapping AD and AR genes. From this, they get overlapping AD and longevity genes. Yeah, by this database overlapping, they get the overlapping AD genes and AR genes. And from this, the reactome analysis of overlapping AD and longevity genes. Yeah, here we have AD genes, that is the reactome pathway enrichment of AD genes. In this, there are three different types of functions. First is metabolism of RNA and also the immune system and transport of small molecules. These were completely based on cascade processes. First one is metabolism of RNA, includes DNA processing, which is transfer RNA, and also the different types of RNA processing, that is ribosomal RNA. It undergoes DNA processing. It is completely based on mitochondria. And also when it comes to immune system, it is completely dependent on different types of interleukins. Example, interleukin 10 and 4 and 13. They helps in signaling pathway. And also the transport of small molecules includes plasma lipoprotein assembly, remodeling, and clearance. And also the plasma lipoprotein clearance. This is a reactome pathway enrichment of Alzheimer disease genes. Yeah, this is for longevity genes. It is also includes the cascade pathways, which is differentiated into signal transduction and gene expression. The signal transduction includes the intracellular signaling by second messengers, that is mTOR signaling, and PIP3 activates AKT signaling. And also from here, gene expression, it includes RNA pol to transcription, and also generic transcription pathway, which is further differentiated into transcriptional regulation by TP53, and also the FOXO-mediated transcription. Yeah, based on the study, the results for the genetic network in this study used that should contribute as a blueprint for the personalized genetic risk assessment for the Alzheimer's disease and other age-related comorbidities. The gene set enrichment analysis, it is a computational method that is useful to interpret the biological functions of gene set with statistical confidence. And also the Reactome, which is a bioinformatics database, it incorporates gene set enrichment analysis embedded within the knowledge database that covers more than 100 bioinformatic resources. The results from the study that represent the most current genetic hallmarks which was involved in Alzheimer's disease, aging, and longevity. Yeah, when it comes to the discussion part, they identified the different types of genetic pathways and perform the balancing between the longevity and aging. According to the image, we can find that there are 3 different types of pathways which was similar in longevity and aging. That is the component immune system, and here we have the impaired immune system, and here we have the intact signal transduction, and here it is impaired. By comparison of these two, longevity and aging, here in aging there are 3 different types of pathways which are unique. They were cellular senescence, protein metabolism, and also the altered gene expression, and also the impaired DNA repair. Yeah, coming to discussion part, the study identifies a diverse range of biochemical pathways which was using by the Reactome analysis of each individual set of Alzheimer's disease genes, age-related genes, and also the longevity genes. The hallmarks of each subset and comparing each individual gene set to the pathways involved in the overlapping of ADAR and AD longevity gene sets. As you can see in the Venn diagram, here it is the Alzheimer's disease genes, and here it is the aging-related genes. From the Venn diagram, we can see that the Alzheimer's disease genes is common, and here the aging-related genes and the longevity genes were variants. And in the middle, we can see these are the overlaps. When it comes to Alzheimer's disease genes, it performs the metabolism of RNA, plasma, lipoprotein assembly, remodeling, and clearance. And also age-related genes, which includes DNA repair, cellular response to stress. And here it is same as this part. And also in longevity genes, it includes signal transduction and also the gene expression. When compared to the overlapping part, it includes gene expression, protein metabolism and simulation, signal transduction, immune system programmed cell death, which is apoptosis, platelet degranulation. And also here immune system, plasma, lipoprotein assembly, remodeling and clearance, and also metabolism of fat soluble vitamins. Yeah, when it comes to the limitation of this article, a potential limitation is that connections among genes and annotations may be influenced by outliers among previous studies, which should be minimized by peer review, systematic review, and meta-analysis. Manually curated annotation nomenclature, some of which may not be easy to understand. The nomenclature which was not easy to understand for the common people. And also the gene set enrichment analysis, it identifies the gene hits within the group of genes in a particular pathway supported by statistical confidence. The statistical confidence and the minimum bias, since a single gene effect is not necessarily predictable, to define the phenotypes and trajectories. Yeah, comes to the conclusion part. In this study, using human gene sets, they have successfully identified and overviewed the gene networks of aging and longevity and the association with Alzheimer's disease genes. And they use the Reactome analysis, which provides the genetic pathway with gene set enrichment analysis and also the statistical confidence levels. The genetic hallmarks identified that provides unexpectedly broad mechanisms suggesting a wide variety of implications in the field of aging. Research such as this study on the genetic network is expected to link aging, midlife common diseases, and Alzheimer's disease. Yeah, when it comes to the impact of the research, the genetic interaction networks among aging, longevity, and Alzheimer's disease, they provide the extraction and translation of gene information into hallmarks as well, as they are the key to developing effects for treatment of Alzheimer's disease. The significant genes involved in these pathways, including TP53, FOXO-SUMylation, interleukins 4, 6, ApoE, and CEPT. These genes suggested mapping the gene network pathways to provide the useful basis for further medical research on Alzheimer's disease and healthy aging. Yeah, thank you for your patience. You can find me at shruti@redix.org.
Thank you so much, Shruti, for the presentation. Now the audience can ask your questions on today's presentation. We have a question, Shruti, here from Vijay. How were longevity genes identified to be impacting longevity?
Okay, yeah. The longevity genes were identified based on the Alzheimer's disease genes, which has a threshold value, that is P, which is greater than 1, based on the genes in humans, which were linked with Alzheimer's genes. Based on the network of this threshold value, they find the longevity genes.
Hope it answers your question, Vijay. And we have another question from Nupur. How the longevity and aging genes were identified from database?
Yeah, they run the Reactome analysis on the app based on the different types of genetic pathways. They identify the overlapping of Alzheimer's disease along with the longevity genes.
Hope it clarifies your question, Nupur. I think there is no more questions, Shruti. Thank you for the presentation.
Yeah.
Can you go for the next slide? Yeah. Before we conclude this webinar, I would like to inform everyone that our next presentation will be on 10 January 2024, titled Longevity Science and Women's Health and Well-Being, and the presenter will be Pratyusha Kupuli. Thank you all for the kind attention and spending your time on today's webinar. Hope to see you all in your next presentation. Thank you.